Aloha everyone, I'm Mark Dugay, a graduate student of Grand Canyon University. In this presentation, we will be learning about glycolysis, gluconeogenesis, and the role of aldolase gene expression. After this teaching learning process, we can explain the glycolysis and gluconeogenesis pathways, explain how aldolase is involved in both glycolysis and gluconeogenesis, describe the different isoforms of aldolase and their different expression patterns, and examine of how aldolase B expression can be regulated by transcription factors HNF1, HNF3, CEPB, and DB. To start with, may we fill out a Venn diagram to compare and contrast glycolysis and gluconeogenesis. So this is just a way to have a quick check on what we know about these two pathways, considering their differences and similarities. This figure shows the opposing pathways of glucose, glycolysis and gluconeogenesis. Let's take a look at first to some of their features that would differentiate between these two metabolic pathways. Starting with glycolysis, it is a catabolic pathway where energy is released by breaking down larger molecules into smaller molecules, from glucose to pyruvate, and it has 10 steps involved. Whereas in gluconeogenesis, it is an anabolic pathway that uses the energy released by catabolic pathways to build larger molecules from smaller molecules. So from pyruvate to glucose, and it has 11 steps. So glycolysis occurs in the cell cytoplasm, whereas gluconeogenesis occurs in the mitochondrion and cytoplasm, where it mostly occurs in the liver. So the raw materials and products of each pathway, where glycolysis starts with one molecule of glucose and ends with two pyruvate molecules and a total of four ATP molecules and two NADH molecules. Whereas in gluconeogenesis, it requires pyruvate and converts it into glucose. So enzymes are catalysts that hasten chemical reaction. Each pathway uniquely involves the following. So for glycolysis, we have exokinase, phosphofructokinase, or the PFK1, and the pyruvate kinase. And gluconeogenesis involves pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6 biphosphate, and glucose 6 phosphate, phosphatase. So for the net reaction of each pathway, so for glycolysis, so these are the reactants. So it needs glucose, 2 NAD plus 2 ADP, and 2 inorganic phosphate. And the products are 2 pyruvate, 2 NADH, and 2 hydrogen ion, and 2 net ATP. While in gluconeogenesis, so the reactants are 2 pyruvate, 2 NADH, 2 hydrogen ion, 4 ATP molecules, 2 GTP molecules, 6 molecules of water, and 2 hydrogen ions to yield glucose and 2 NAD and 4 NADP plus 2 GDP and 6 inorganic phosphate. So the breakdown of 6 carbon glucose molecule into two 3 carbon pyruvate molecules occurs in 10 steps, which is further divided into two phases. So first is the preparatory phase or the energy investment phase, which constitutes the first five glycolytic steps. And second is the payoff phase or the energy yielding phase, which constitutes the last five glycolytic steps. So step one, glucose enters the cell and is phosphorylated at the hydroxyl group of carbon 6 catalyzed by the enzyme hexokinase, requiring magnesium ion for its activity to transfer a phosphate group from ATP forming glucose 6 phosphate. Phosphorylation of glucose ensures that pathway intermediates remain in the cell. Step 2, the reversible isomerization of glucose 6 phosphate and aldose is catalyzed by the enzyme phosphohexose isomerase to yield fructose 6 phosphate, a ketose. Step 3. In this step, still another ATP molecule is invested in glycolysis. The transfer of a phosphoryl group from ATP to fructose 6 phosphate is catalyzed by the enzyme phosphofructokinase 1 or the PFK1 to yield fructose 1,6 biphosphate. With phosphate groups on its opposite ends, the sugar is now ready to be split in half. The activity of PFK1 is regulated by allosteric mechanisms, whereby its activity is reduced when cells have high ATP levels. Step 4, cleavage of fructose 1,6 biphosphate by enzyme aldolase to yield to different trisphosphates, specifically the dihydroxyacetone phosphate, a ketose, and a glyceraldehyde 3-phosphate and aldose. Step 5, interconversion of the trisephosphates. The two different trisephosphates have different fates, whereby the dihydroxyacetone phosphate is rapidly and reversibly converted to glyceraldehyde 3-phosphate by the fifth enzyme of the glycolytic sequence, triphosphate isomerase. Okay, whereas glyceraldehyde 3-phosphate can be directly degraded in the subsequent steps of glycolysis. This reaction completes the preparatory phase of glycolysis. The hexose molecule has been phosphorylated at carbon 1 and the carbon 6, and then cleaved to form two molecules of glyceraldehyde. So the second phase is basically the payoff phase or the energy yielding phase, which constitutes the last five glycolytic steps. Some of the chemical energy of glucose is converted in the form of ATP and NADH. Step 6, the first step of payoff phase, where the oxidation of glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate with the presence of glyceraldehyde 3-phosphate dehydrogenase. This step is one of the energy conserving reactions of glycolysis where one molecule of NADH is released. Step 7. The reaction shows a substrate level phosphorylation in which phosphoryl group is transferred from 1,3-biphosphoglycerate to ADP catalyzed by phosphoglycerate kinase yielding 3-phosphoglycerate and ATP. The first two steps, steps 6 and 7 in the second glycolytic phase, create an energy coupling process in which 1,3-biphosphoglycerate is the common intermediate. The outcome of these coupled reactions were both reversible under cellular condition, that the energy is released on oxidation of an aldehyde to a carboxylate group is conserved by the coupled formation of ATP from ADP and inorganic phosphate. Step 8 is the conversion of 3-phosphoglycerate to 2-phosphoglycerate phosphoglycerate catalyzed by phosphoglycerate mutase. The enzyme causes a reversible shift of the phosphoryl group between carbon 2 and carbon 3 of phosphoglycerate. In this reaction, magnesium ion plays an essential role as a cofactor of the enzyme to transfer a phosphate group to or from a molecule, plus it helps to shield the negative charges from the phosphate groups of ATP to allow the enzyme to properly function. Okay, step 9, the enzyme enolase catalyzes 2-phosphoglycerate to phosphoenol pyruvate and promotes reversible removal of a molecule of water. Okay, step 10. This reaction is another substrate level phosphorylation in which phosphoryl group is transferred from phosphoenol pyruvate or PEP to add to ADP catalyzed by pyruvate kinase with the presence of potassium and magnesium ions yielding ATP in the end product pyruvate. So this is the glycolysis pathway involving the 10 different steps. Okay, table 1 shows the sequential reactions in gluconeogenesis, starting from pyruvate to glucose. Gluconeogenesis involves non-carbohydrate precursors for glucose biosynthesis and maintains the normal glucose level and energy requirements needed by the body cells, particularly the neurons and erythrocytes. Step 1 in gluconeogenesis is the conversion of pyruvate to oxaloacetate, catalyzed by the enzyme pyruvate carboxylase in the mitochondrion where ATP molecule is used. Okay, step 2, conversion of oxaloacetate to phosphoenol pyruvate, catalyzed by the enzyme phosphoenol pyruvate carboxykinase or the PEPCK using GTP molecule. Prior to this reaction, oxaloacetate is shuttled into the cytosol from the mitochondrion. Oxaloacetate consumes NADH to be reduced to malate in order to leave the mitochondrion and reoxidized back to oxaloacetate once it is in the cytosol releasing NADH. Okay, step 3 through 8 are reverse reactions of glycolysis catalyzed by the same glycolytic enzymes, where these steps do not require energy. Okay, step 9 is a hydrolysis reaction and is irreversible, where fructose 1,6 biphosphate is converted to fructose 6 phosphate catalyzed by fructose 1,6 biphosphatase. Okay, step 10, reverse of glycolysis where glucose 6 phosphate is produced. And the last step is another hydrolysis reaction and irreversible where glucose 6 phosphate is converted to glucose catalyzed by glucose 6 phosphatase, which is transported into the endoplasmic reticulum by glucose transporter 7, which is only found in liver, kidney, pancreas, and small intestine. So this presents the gluconeogenesis pathway. So how aldolase is involved in both glycolysis and gluconeogenesis? Aldolase is involved by glucose phosphorylation and isomerization. Aldolase on the enzymes involved in both is an enzyme involved in both glycolysis and gluconeogenesis by glucose phosphorylation and isomerization. In glycolysis, aldolase catalyzes breakdown of fructose 1,6-biphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate occurring in the fourth step. Whereas in gluconeogenesis, a reversing reaction occurs where aldolase catalyzes the synthesis of fructose 1,6-biphosphate from this triose phosphate, the dihydroxyacetone phosphate, and the glyceraldehyde 3-phosphate. So isoforms of aldolases are abundant in the human body and play roles in glycolysis, gluconeogenesis, and even in fructolysis and other metabolic pathways. There are three genetically distinct and tissue-specific isozymes of fructose biphosphate aldolase, class 1 in mammals. We have here aldolase A, aldolase B, and aldolase C. All the three aldolase isozymes catalyze the reversible cleavage of fructose 1,6-biphosphate or fructose 1-phosphate to dihydroxyacetone phosphate and either glyceraldehyde 3-phosphate or glyceraldehyde, respectively. The aldolase A is expressed mainly in muscle. Developing embryos produces aldolase A, which continues to be expressed in many adult tissues, sometimes at much higher levels than in embryo. In adult muscle, aldolase A can be as much as 5% of total cellular protein involved in the action potential, which allows movement or locomotion. Aldolase B is expressed in the liver, kidney, stomach, and intestine, whereby it is the aldolase B is the only expressed isoform in highly differentiated hepatocytes. The high level of gene expression results from cooperation between a liver-specific promoter and an intronic enhancer. And last, the aldolase is expressed in the brain, heart, and ovary. The aldolase B isozyme uh, possess, possesses a crucial role in metabolic pathways, particularly in glycolysis and gluconeogenesis. So this figure shows the map of the promoter of genes coding for glycolytic enzymes. We'll be focusing on enzyme aldolase B. Its gene expression appeared regulated to the proximal tubule, the site where hepatocyte nuclear factor 1 or the HNF1, a hepatocyte nuclear factor 3 or the HNF3, we have the C enhancer binding protein or the CEBP and albumin D binding protein transcripts. So figure 5 shows the liver-enriched transcription factors that regulate genes coding for glycolytic and gluconeogenic enzymes. Aldolase B promoters, which contain the different transcription factors such as HNF1, which is an activator, HNF3, which is also an activator, and another transcription activator is the DBP. And the last transcription factor, which is a regulator, is the CEBP. So a study conducted by Gregory on the competition between the transcription factors HNF1 and HNF3 and alternative cell-specific activation of DBP and CEBP contribute to the regulation of the liver-specific aldolase B promoter, show that in HepG2 cells, HNF1 and DBP are strong activators of aldolase B, which increases the gene expression, whereas the CEBP was found to weakly activate aldolase B in HepG2 cells and strongly activate aldolase B in hepatocytes. The CEBP is considered as the central regulator of energy metabolism because it regulates gene coding for enzymes that control energy processes in tissues involved in uptake, metabolism, and storage of physiological fuels. Okay, so we're down to our summary. So, metabolic pathways include glycolysis and gluconeogenesis. Glycolysis is a catabolic pathway that breaks glucose to pyruvate with a net yield of 2 ATP molecules and NADH per glucose molecule used. While gluconeogenesis is an anabolic pathway, the biosynthesis of glucose from non-carbohydrate precursors. Enzyme aldolase involved in both glycolysis and gluconeogenesis by glucose phosphorylation and isomerization, specifically converting fructose 1,6 biphosphate into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate in glycolysis and its reverse reaction for gluconeogenesis. Aldolase has three isoforms. We have aldolase A, aldolase B, and aldolase C, which are expressed in muscles, liver, and brain, respectively. And the expression of aldolase B can be regulated by the transcription activators such as hepatocyte nuclear factor 1, hepatocyte nuclear factor 3, and albumin D binding protein, and the transcription regulator, the C enhancer binding protein. So again, to check for understanding on the topic presented, I would like you to go over your Venn diagram you had earlier and write what you learned about the differences and similarities between glycolysis and gluconeogenesis, considering their pathways, reactions, enzymes involved, and the like. Okay, so that ends my presentation. Thank you for listening. Mahalo.